Okay, thank you for your kind introduction. And it is my great honor to have this opportunity to introduce our work. And this is a outline of my presentation. I will talk each one by one. As we know, proton amount and proton activity are widely measured in the field of diagnostics and biopharmaceuticals. And the proton amount means the number of proton molecules defined by its primary sequence. The proton activity means the function of the specific proton. In order to set up the traceability hierarchy for proton amount and proton activity measurement, we need to develop metrological methods to quantify proton amount as well as activity accurately, especially for biologically active proton connotation. This slide shows the proton connotation issue. Let's assume the light blue indicates the active proton molecules, and the dark blue indicates the inactive proton molecules. Other colors indicate the proton impurities and non-proton impurities. So the proton connotation issues can be classified as following. Firstly, if we count all the proton particles together, I mean, if we count inactive proton particles, active proton particles, and proton impurities together, that will be total proton connotation. Otherwise, if we just count inactive proton particles and active proton particles, that will generally called as proton connotation by its primary sequence. In this way, we do not differ the inactive protons from active protons. However, if we just want to count the active proton particles, that will be active proton connotation. And the active proton should be defined before the measurement. Generally, the active proton can be defined by its specific structure or activity or function. In order to measure the proton amount or proton activity accurately, we need a primary method. A primary method is the method having the highest metrological qualities, whose operation can be completely described and understood, for which a complete uncertainty statement can be written down in terms of SI unit. And the CCQM has identified several potential primary methods. For example, isotope dilution mass spectrometry, and coulometry, gravimetry, tetrametry, and collective methods. However, among these methods, I think only isotope dilution mass spectrometry can be used for proton amount measurement. In order to find more potential primary methods for proton amount and proton activity measurement, NIM China has a plan to study on the new potential primary methods in recent years. For proton amount measurement, we plan to set up LC-CD method, IDSUS method, the single molecular counting method, as well as ESDMA CPC method. And for active proton connotation, we plan to develop SPR method, digital ELISA method, and the digital PCR-based proton connotation methods. Now we have made some progress on LC-CD method and SPR method. So in this presentation, I will talk about these two methods. First is the HPLC-CD method used for proton amount measurement. As we know, circular decolorism is a decolorism involving circularly polarized light, the differential absorption of left and right-hand light. Generally, a pair of the chiral compound have a mirrored CD spectrum. That means they give the equal but opposite direction CD signals, like the picture showing. This is the CD spectrum of alphenylidene. And this is the CD spectrum of D-phenylenediene. And if we mix the alphenylidene with D-phenylenediene with the same amount, we will get a racemate solution. And the racemate solution has no CD signal. So in this way, the CD is just like a balance. So we can use D-type of amino acid as standards to quantify L-amino acid in the samples directly. And if the balance is equilibrium, that means the amount of L-amino acid in the sample is equal to the amount of D-amino acid in the standards. We can also set up internal standard method. In this way, we add D-type of amino acid into both samples and standards. And if the two balance are equilibrium, we can know the amount of L-amino acid in the sample is equal to the amount of L-amino acid in the standards. If we use HPLC-CD with UV and CD detectors, we can detect the peak in UV channel. However, if the amount is the same in the CD channel, we cannot observe the CD signal. So, in order to validate our theory, firstly, we should set up the LC-CD instrument. We connect commercial Agilent HPLC with commercial JASCO CD spectrometer, like this. After that, we select alphenylidene as an example to validate our theory. Firstly, we optimize the detection and separation conditions for alphenylidene. We try to separate alphenylidene from other 16 amino acid mixtures. Then we can use D-phenylenediene to quantify alphenylidene in the mixture. Firstly, we just use CD spectrometer to validate the theory. In this approach, we use only pure alphenylidene solutions prepared by certified reference materials. And in this way, the optimal range is from 0.04 to 0.8 milligram per gram. In this optimal range, both the Bayes and the relative standard deviation are smaller than 1%. Then we use HPLC-CD to separate alphenylidene from the mixtures and to validate the theory. In this approach, the optimal range is from 0.5 to 1 milligram per gram. So this optimal range is very narrow. However, in this narrow range, both Bayes and relative standard deviation are smaller than 1%. Then we use this technology to participate in CCQM K78 international comparisons. We use HPLC-CD to quantify 4 amino acids in the samples. That is proline, leucine, isoleucine, and phenylenediene. We use a direct analysis approach as well as internal standard approach to quantify alphenylidene in the samples. The result is shown here. The gravimetric value is provided by the coordinate laboratories. And the middle is the result obtained by IDMS technology and reported by NIM China. And the last one is the result obtained by HPLC-CD technology. We can see the uncertainty obtained by HPLC-CD technology is much smaller compared with that obtained by IDMS technology. And the Bayes was only about 0.16%. We have also tried this technology for leucine, isoleucine, and proline connotation. However, because the concentration is lower than the optimal range, concentration range, and due to the separation problem, the results of these 3 amino acids are not as good as that of phenolaniline. As we know, natural proteins consist of only L type of amino acid. So we can hydrolyze the protein into amino acid mixtures. Then we can use D type of amino acid to quantify L type of amino acid in the hydrolyzed state. Then we can use amino acid concentration to calculate the protein amount. To do this, firstly, we should optimize the separation conditions to ensure L-phenylaniline can be separated from the hydrolyzed state. Then we have tried this technology for pulsing insulin, G2 EPS-PS protein, and human myoglobin purity analysis. We can see the result obtained by LC-CD technology are comparable with those from LC-IDMS technology. And the EN value are smaller than 1. And the uncertainty obtained by LC-CD is smaller than that obtained by LC-IDMS technology. However, the decrease is not obvious because the hydrolysis process still contributes a large uncertainty components. We have compared the HPLC-CD method with traditional HPLC-IDMS method, and we can see a lot of similarities. So we think HPLC-CD could be a potential primary method for connotation. However, the disadvantage for LC-CD is for its low sensitivity. And this method needs complete separation of target from the interferences. The potential application for this method can be used for amino acid analysis, protein purity analysis, and maybe it can be used for natural product, compound, traditional Chinese medicine analysis. Okay, the second method is SPR-based active protein concentration measurement. This is a measurement per sample for SPR. This is the light source, and the right is the detector. And the antibody are immobilized on the surface of the sensor chip. When the proteins, the antigen, pass through the flow channel, it can be captured by the antibody. So a linear relationship is found between the resonance energy and the mass concentration of biochemically relevant molecules. So if the antibody catches the antigens, we can see the increase of the signal. And if the complex dissociates, we can see the decrease of the signal. And if we remove the antigens from the antibodies, the signal comes to the baseline. So this process is called regeneration. And this is a measurement per sample of calibration-free concentration analysis by SPR. And I think this measurement per sample is originally proposed by GE company. The protein in the solution can come to the surface and captured by its antibody. So based on this process, we can write this equation. And based on this equation, we can draw the second equation like this. The left is the binding rate, and the right is the constant plus the concentration. So if we know the binding rate and we know the constant, we can calculate the concentration directly without any external standard. So this will be an absolute quantitation method. We can use SPR to determine the binding rate directly. And GE has provided software to estimate the value of the constant. So with the binding rate and the constant, we can calculate the concentration directly. And this concentration stands for the proteins that could be captured by its antibodies. So this is some kind of active concentration. To do this, firstly, we should optimize the antibody immobilization conditions, as well as regeneration conditions. After that, we should optimize the immobilization levels, and we can see with the increase of the immobilization levels, the active concentration obtained is converged, and the relative standard deviation becomes smaller. Then, with the optimal experimental conditions, we have analyzed the active concentration in 3 separate days. The relative standard deviation within 1 day is from 2.5 to 4.6%. And the relative standard deviation among 3 days is about 8.4%. Then, when we have the active concentration, we can compare this result with the proton amount obtained by IDMS technology. So we can calculate the active fraction. We have tried this technology for G2 EPSPS proton and for human myoglobin. For G2 EPSPS proton, the active fraction is about 18% for the given antibody. However, for the human myoglobin and the given antibody, the active fraction is about 43%. So when we have the active concentrations, we can use this data to accurately characterize the interaction between antigen and antibodies. This is the example. We use active concentration to calculate the kinetic and thermodynamic constant between G2 proton and its antibody. And the data is shown here. They are significantly different from those obtained. Just use the proton amount concentration to calculate. So using the active concentration, we can get much reliable molecular interactions. So finally, I'd like to thank Luo Yi, He Zhangjing, Zheng Kangle, Su Ping, and Yang Yi for this work, and I'd like to thank AQSIQ for financial support. Finally, I'd like to make a summary. LC-CD was established to quantify amino acids and proteins in solution or solid form, showing better repeatability and lower uncertainty compared with IDMS. SPR-based CFC method was developed to quantify the active concentration of proteins with specific immunoactivity without any external standards. Both proposed methods can be entirely described by equations and could be potential primary methods for both proton amount and activity measurement. That's all. Thank you.